Welcome to Akhand Vidyashram, the first institute of impeccable wisdom. The perfect human cycle, the divine design of human cycle, my dharma, the impeccable wisdom, my greatest discovery, Devyank, the divine design of nature with which the perfect objects of the universe are designed and created. Impeccable wisdom is enlightening, Akhand Vidya wisdom is empowering. With Devyank, the first time revealed in 2015, we are going to see the entire universe, human body, and every aspect of human body in that light and say how beautiful is the core, how we should maintain that for rest of life and live a comfortably healthy, wealthy, wise and happy life for generations to come. The human cell, as you know, is the minimum independent functional unit of the body. There are 100 trillion cells which are required to make body as we are. There are different types of cells, 200 different types, but there are some common characteristics which we are going to talk about the cell, particularly the living cell, which can multiply and grow further. As I've been saying very clearly, the universe is one, everything could be one. But when it comes to the manifestation, there are always three parts. And as far as a human cell is concerned, it's also got three parts. Under the compound microscope, we can see three parts as the first one, you can see right on top, cell membrane, which encloses the cell and separates it from the extracellular fluid and intracellular fluid. Exchange is difficult because this cell permits lipophilic things to go through, but hydrophilic things cannot go through. As a result, the cell size, shape of the cell can be maintained because of cell membrane. Second part is cytoplasm, and third part is the nucleus. These are three independent parts which play a very functional role as such, and we are going to understand those roles and see how beautiful is the divine design of independent anatomical and functional unit of the human body. Now let's say human cell under the electron microscope are electrified. You have 21 parts. That means cell membrane is made of 7 parts, cytoplasm is made of 7 organelles, 7 parts, nucleus is also made of 7 parts, total there are 21. As I've been saying over and over, there is one part which is very close to the nucleus. Sometimes people consider it cytoplasm part, some people, I call it as the nucleus part. If it is included as the nucleus part, also is fair enough because it functions along with the nucleus in division. It may be outside just very close to that. It's not very far away from the nucleus. That is something, and this is the one called centrosome, which has got two centrioles inside together in a unit, enclosed unit, and that has to be separated during first division of this. As a result, 21 part becomes 22 part, as the first division of cell is 22 by 21. I have been saying that from the day one. This is a great enlightening knowledge for me. Now come to the cell membrane. Let's understand that cell membrane has got 7 important parts. This picture is from Encyclopedia Britannica, and I consider their structures a bit. Encyclopedia Britannica is much better, and whenever I find their structure with their permission, with their knowledge, with their logo, I'm saying that you have this particular structure well defined, suits my objective better. Now you can see on the left side you have outer surface as well as inner surface, and of course outer surface is exposed to extracellular fluid and inner surface is exposed to intracellular fluid. But there are two different layers which are called lipid bilayers, and they primarily are lipid in nature, which prevent the easy exchange from extracellular fluid to intracellular fluid. The things were in a solution form or water form. But as far as fatty acids are concerned, they can easily go through. So first part is lipid, two layers of cell membrane you can see on the left side. Then there is something which I want to emphasize on, hydrophilic polar head of the phosphoric acids. This lipid which makes two layers, one outermost part, both extracellular fluid and intracellular fluid is phosphoric end, and inside is hydro non-polar hydrophobic fatty acid tail. That is something which has to be understood why they are different, so that things cannot pass through and cell is protected, is a big shield by itself. Third thing, cholesterol layer of the cell membrane is also important because cholesterol is the raw material for all the steroids, and that is required for exchange of fatty acids or steroids or cholesterol, anything from outside to inside. But as far as solution is concerned, water is concerned, amino acids are concerned, carbohydrates are concerned, they need to go through certain passages which are important. What are those passages? First thing, you have sugar side chain of the cell membrane, which helps you to bring sugar in. It could have a direct sugar, or along with the sodium is carried along through the pores. Then you have intrinsic protein long chains, which help most of the things to go inside, and they work through sodium potassium pump, ATP pump. And then you also have extrinsic protein short chain as the intrinsic part short chain. So these 7 parts play a very important role to maintain health of the cells perfectly, beautifully, protected from all the damage outside, all the changes outside do not affect the cell core as such, and cell can function as optimally as possible because it's protected. If you drink too much of water, there could be dilution, but the water will not reach the cell and change it. It's protected. Or if you take too much of sugar, you take whatever else, that is maintained outside in extracellular fluid, interstitial fluid, blood and otherwise, but doesn't go into the cell unless until there is a damage to the cell membrane. So that way, I think I made it very clear. Cell membrane is a shield, is a protection, is a structure checker like your bones and muscles outside. For the cell, you can call this also as mesoskeleton part, or the part which play important in shield like muscles and bones. Now to explain two lipid layers, let's say extracellular fluid has got all the molecules you can see, all the elements you can see. They can go through only sodium potassium pump as you can see. Right on the left side, it is there, nothing can go in. But unless until the membrane opens, then only elements can go in, whether it's a sodium, potassium or otherwise. It's as simple as that. But then after that is closed with a pump, electrogenic. As a result, potassium from inside cannot come out and sodium cannot come down. As a result, extracellular fluid concentration, electronic concentration, and intracellular electronic concentration is maintained to 238 or 230 milliequivalents. That's the most beautiful part. As long as there is no damage to the cell membrane, no damage to lipid layer, we can lead a perfectly healthy life. So this is something which I always want every student of mine to understand, how beautifully we are protected. But everything is mathematically based and science and put together. For that, sodium and chloride ratio of protons is also in deviant ratio. Sodium is 11 and chloride is 17. That is it. But sodium and potassium also cannot work against each other. Sodium is very powerful. Potassium is not that powerful. But for them to exchange from inside, outside exchange, you need ATP pump or sodium potassium ATP pump. That's such a beautiful science. And that also to maintain intrinsic factor. Nernst equation comes to play a role, and that got a log of 61, and I do believe it should be 61.8034 if you want to have a perfect equilibrium. This is also something which I have spoken about it we are going to talk about all the deviant in everywhere in the body, how if we can maintain that through the intake also and maintain the exchange perfect, we could reverse aging itself. And I'm trying to do that in my life. Even at 73, I'm trying to maintain my biochemistry as perfect as I was in 21 without any difficulty of any nature. Now come to the human cell, you have 7 organelles. And those organelles are, first I'm talking about mitochondria, which is energy molecule producing ATP, which is required for the exchange of glucose, proteins, sodium, potassium, electrolytes, and their exchange, all the important things go through that as a second transport system. Passive is one which happens for higher concentration to lower concentration, does not need ATP. But otherwise, which cannot go through easily, you need ATP playing a very important role, and that is something which I want everybody to understand perfectly. Second thing is rough endoplasmic reticulum, which is surrounding the nucleus. Then you have smooth endoplasmic reticulum. Then you have Golgi body apparatus. Then you have lysosome, ribosome, and I have put centrosome here just to highlight it as one part, which could also be vesicles, and centrosome could be a part of the nucleus. That's why in the last I put it at the borderline, which is very close to nucleus. That's it. Let's understand mitochondria. This is the structure of mitochondria, beautifully seen. You can see the outer membrane, you can see the inner membrane, you can see intermediate space, you can see the matrix, you can also see DNA, and you can also see ATPase being formed inside the way you can see F of portion 1, F1 portion 2. And this is also something you have inside as a ribosome, which are inside to produce ATP, also ribosomes outside. That's also something we are going to discuss about. But here one point which I want to make clear, nucleus is, by mitochondria, is derived from Escherichia coli E. coli cells. What was working in DNA of E. coli is here. But why I'm talking about E. coli? E. coli is one of the cells which can be multiplied 10 times. After that, it comes to an end. It's so beautifully seen. Same thing must be here too as far as ATP is concerned. So cells must be regenerating to produce DNA of its kind, which is called molecular DNA or mitochondrial DNA. Then comes to endoplasmic reticulum, which has a two-part, rough endoplasmic reticulum, which is closed, and then you have smooth reticulum. But rough endoplasmic is responsible for synthesis of proteins, and that is based on DNA structure inside the nucleus. When they come out, convert DNA into RNA, and then post-translational thing takes place, modification, folding, sorting took place, all in endoplasmic reticulum only. But smooth endoplasmic reticulum is responsible for synthesis of lipids. Protein is by rough, lipid is by this. That's very interesting. And here you have the synthesis of lipids like phospholipids, cholesterol molecules which are essential. It is also responsible for creation of steroids and metabolism of carbohydrate at a later date. That is also something which need to be understood, is a beautiful part of endoplasmic cells. As a result, cells can produce, they are chemical industry. They can do wonders. Provided we get a good quality raw material to the cells, you can make cells survive and reverse aging also over a period, depending upon how beautiful is our food intake, how is the vegetarian diet. We do not take anything which is damaging, no alcohol, no smoking, no drugs, anything which is unnatural should be avoided. What is natural should be given in the right concentration. That all is again designed with Divyank alone. Then comes to the Golgi body apparatus. And this is something which is closed. It transports the molecules synthesized by rough endoplasmic reticulum. It's very clear. Rough endoplasmic reticulum is responsible for amino acid and proteins. The molecules faced with the membrane are sorted out for destination. Molecules undergo remodeling and modification in the system. The molecules are secreted out from the cell to be used later by other organelles like your ribosome. If it is protein, it's a ribosome. If it's cells to be broken down, it's a lysosome. That is something very interesting. The sequence we can see is mathematical design, and it's also again Deviank. As a lysosome is concerned, as you can see the picture very clearly, there's a larger organelles with a membrane. There's a membrane in this. So, whatever changes are going to take place is inside the cell. And it can synthesize hydrogen peroxide for lysis. It breaks down all the waste products as well as cellular products which are not needed. You have infection, you have things. All those things will easily be broken down with the hydrogen peroxide and important. So what a wonderful way of nature design. Whatever is required is produced locally and does its wonders within the cell. Then comes the ribosome is the structure which I have already explained, which I am going to explain again in the formation of amino acids and formation of proteins based on RNA, DNA structure. I have already done, we will be doing more in detail because every new one has to be better than what I have done earlier. So that the sequence is maintained, everybody has the impeccable wisdom, etc. It is not possible for me to give you all the wisdom in 20 minutes or 22 minutes. So it's going to be destroyed again. But having understood the first part, the next in that series would be at least another 25 to 30% more, then further another 30% more. The more and more from first you start watching my videos, you will find at the end, maybe after 4 years, 5 years, or 10 years, I do not know when I'll be able to complete. You will have all the wisdom in your hands, A to Z of the entire mathematics, divine mathematics, physics, chemistry, perfect laws, anatomy, physiology, biochemistry, psychology at your fingertips. You'll have all the knowledge. Your brain will be one with the universal consciousness. And if you follow all what I've recommended, I can assure you, you could be as vibrant, as dynamic, even in your 70s, 80s, 90s, 100s. You will remain young forever. Reverse of aging is required, and you for that only knowledge is required, and that is the knowledge I am organizing, understanding, implying enlightenment, and become an example. And I'm sharing everything based on my example. It's not a copy paste. It is not a copy of something else, but science in the light of Deviank. Now comes the centrosome and centrioles. This is a beautiful picture of two centrioles as a centrosome on the left side, and also the structure on the side which is electronic. This is very much like the structure you can see on top of both the sides and top. They are identical. They are the 9th level of Pragyan. Pragyan plays a role in creation. You can see the pictures on either side. Amazing. And there are two closely held segments of the centrosome as shown in the right picture. And also during the first stage of creation, these two segments separate and become two centrioles. Centrioles move to the opposite side. They are held to the mitotic spindles of the cell. Another centriole, there is one mother centriole, one daughter centriole, and one remains, and only one centriole is passed on to the next cell. It regulates the division of the cell, and that is what makes 21 parts as 22 parts. And the first stage of creation is 22, 21 is primarily because of centrosome. It is not because of nucleus, not because of anything else. Whereas the book gives you knowledge that is nucleus is required, but actually it's a centrosome, not the nucleus, which becomes two centriole for multiplication in a living cell. And that example I have given is what? A red blood cell which are mature formed. They cannot multiply because there is no nucleus, agree, but there is no centrosome. But also I have given an example of neurons have nucleus. It can generate proteins and other things to maintain the cells for the rest of your life. They do not multiply because the centrosome is missing. That is the kind of knowledge which we need. Now there comes the nucleus cells. As I told you, there are 7 cell part in this also. You can see on the top picture, you have nucleus membrane, you have nuclear pores, you have chromatin segment, you have granules, you have nucleus, you have cryptosomes, chromatin diffuse, and fibers, etc. inside. But the picture down I have shown, you can see centrosome is so closely held to the nucleus itself. And just to show that this makes it 7 part, and you can see microtubules which separate and then divide into two is so closely held. And this information is very precise. I don't think I can do justice in this particular thing in 22 minutes to give you the wisdom of human cell, which we'll take it up later. Now, entire information which I have told is in this particular picture again. Just as a revision of what I've spoken about, cells, there are 100 trillion cells. Their structure and function units of the body. There are different types of cells, but basic structure is what I've seen. Under a compound microscope, you can only see 3 parts. Under electron microscope or higher magnification, you can see 21 parts. Cell membrane, 7 parts. Cytoplasm, 7 organelles. And nucleus, 7 parts. And centrosome becoming 2 and leading 21 parts to 22 parts during first stage of creation is what it is all about. And the examples of further development you can see in RBC cells, because germ cells original are 21 microns which become 22 microns, and they go through 10 stages of development to become mature. That is a beautiful example, and I have already shown, but here I can actually revise you. Remember that picture and do this, and you'll have a perfect wisdom coming up. So 22 by 21 and to the power of 10, I think I've very well done within this, and I think that should be enough for the day. Thank you very much. Thank you.